All right, this lesson is about proteins and it is subtopic 2.4. All right, we are having a look at all of our syllabus points. There's quite a lot here. Proteins are a massive part of the body, so there's a lot going on here, and we will be doing some pracs as well. Okay, proteins are made up of amino acids. They are the monomer, and so there is our amino acid. Remembering that we have a carbon in the middle, a nitrogen group off to the left, which is called the amine group, the nitrogen and two hydrogens, and we have a carboxyl group off to the right there, and the R is that variant. Now, once those amino acids start to join together, it starts to form a long chain, which is known as a polypeptide, right? Makes sense. And once that chain is folded around on itself, then it becomes the protein. So amino acids are the monomer, and then it becomes that polymer, and then once it is folded and possibly added to other polypeptides, we have a protein. All right, there is the condensation reaction right there, which occurs between amino acids to join them together. And you can see here, this is where the action happens between the carbon in the carboxyl group, and it joins to the nitrogen in the amine group, and it kicks off two hydrogens and an oxygen. So you can actually relate that back to here. So it's going to kick off one of these, and it's going to kick off this hydrogen here. Sorry, and this oxygen. And between them, it becomes a peptide bond, right? The bond between them is called a peptide bond. Now, one of your skills in your syllabus is that you need to be able to draw this reaction. So the condensation reaction, which causes that peptide bond, it's very important to be able to represent that in a molecular way. So remembering that this peptide bond is going to be between that carbon sitting there, this stuff is kicked off, one of these hydrogens is removed, and the nitrogen from the amino group is joined in there. Right, so amino acids become proteins. Obviously, they are the monomer. When they are joined together, and there's only two of them, it is called a dipeptide. There's that di, or the di for the prefix meaning two. And we've got an oligopeptide when there is less than 20 amino acids in that chain. And once it goes over 20, we have a polypeptide. So over 20 amino acids there. Once that polypeptide is created, it can be folded, and it can, link in with itself and other chains as well to create a protein. This is insulin. It is quite a simple protein. It's only got two polypeptide chains that link in together. One is 21 amino acids long, and the other one is 30 amino acids long. However, the flip side of that is this is titin. It is a protein in muscle. It is 34,350 amino acids long, and this is its chemical formula. It's quite ridiculous. So we're talking really simple chains there mixing in to huge numbers of them. Now, there are 20 kinds of amino acids. No, you don't have to learn them all, but we're saying it's got the same base structure, and that R group allows it to be a different variant. So the essential amino acids in your body must be taken in via your diet. The non-essential amino acids can be constructed by the body in a sort of Lego fashion. It uses available atoms to construct them. Now, it's the combinations of these. If you have 20 of them, you can combine them in so many different ways. You can have long chains, short chains, and then on top of that, you're then taking other chains and mixing them in. So the number of proteins that are possible, massive, quite ridiculous in fact. But it's all starting from that amino acid, that really base, simple structure right there. Now, genes in the DNA code for proteins. They are the recipe for proteins. That is literally their only job. Every 3 nucleotides along a DNA strand, so 1, 2, 3, 1, 2, 3, and we sort of talked about this a little bit, what codes for one amino acid. So every 3 nucleotides is read as what's known as a codon, C-O-D-O-N, and determines which specific amino acid is next in the chain. So the final protein is made up from a folded polypeptide chain, and there's 4 different structures within that. So 4 different particular ways of that folding can cause a new protein to occur, and the way in which they interact with it themselves even is really important to this. All right, collagen is an example of a protein, and it is 3 polypeptide chains, and they're wound like a rope. Those ropes then wind with each other as well, and they can stretch. They're highly tensile and very, very strong. They're found in connective tissue, which is like the gristle in meat. It holds all your organs together and stuff. You often hear about collagen fillers in your face because it's all about structural integrity inside the body, keeping things nice and perky. All right, this is hemoglobin. Hemoglobin transport oxygen and carbon dioxide around the bloodstream and diffuses those gases into the cells. It's got 4 subunits, okay? Collagen had 3, hemoglobin has 4 polypeptides, and in between they have this heme molecule as well. So there's 4 heme molecules per hemoglobin. In that, so you can see right in the middle of that, there's an iron atom. So if you are anemic, you are not getting enough iron, sometimes your hemoglobin can't be made, and therefore you'll feel fatigued because your gas can't actually transport around your body very well. Protein structure, again, we're back to this again because structure is so important for function. The primary structure is that specific sequence of amino acids. The secondary structure of a protein is the shape the polypeptide makes. So we can see here in the secondary, it's starting to fold in its own particular way. The tertiary structure is the shape of the polypeptide caused by more folding in and around on itself. And the quaternary structure is when we start adding in the other subunits and anything else, like, for example, in the hemoglobin, that heme molecule. Now, structure and function, as I keep saying, go hand in hand. And when we talk about a long protein, so like that collagen, that is folded in one particular way versus the globular proteins like the hemoglobin. So there are also chaperone proteins. Oddly, proteins help to make proteins. And they help, once the ribosome makes the polypeptide chain, they start folding in on itself. So these R groups that stick onto the end of the amino acid, they interact with one another and they bond to stabilize that protein. And that means hydrophobic regions that are not enjoying water go right inside and hide away, and the hydrophilic parts can stay on the outside. So that folding is incredibly specific to make sure it is comfortable in its surroundings or it can dissolve and things like that. In the longer proteins, like the collagen, the R groups purposely don't bond to ensure that it remains elongated. All right, here's a whole bunch of functions. Now, you could spend all day doing this. And here are just 6 examples that the syllabus specifically recommends. There are so many proteins in your body doing so many jobs. We can't possibly learn them all, but we need to have a think about some of these. Denaturation is the process where the protein actually unfolds, and you don't want that to happen, right? It can be caused by heat. Cooking things is pretty much our number one example of denaturing something. When we see a change of an irreversible change of color or texture, all those kinds of things. Now, acid can also do that, and if you've ever heard of a ceviche is a cooking technique often used in seafood where you put it in citrus, so you're putting it in an acid because pH changes those proteins as well. A proteome is like your genome, right? Your genome is your organism's complete set of genes. Your proteome is your complete set of proteins, okay? It's unique, definitely unique to you, but it is variable and can change, right? It's going to change depending on what you need at what time. For example, your proteomes are going to change when you hit puberty and you're producing a lot more hormones, which are proteins. So your proteome is actually much larger than your genome. So even though your recipe book of DNA is saying, right, here's what we can make, once that polypeptide chain is made, those modifications can happen after the fact. So you might have a gene coded for one protein and you could get 5 modifications from that. All right, here are your syllabus points, a lot going on, so please reread over them.